All right, so my topic is neural prosthetics, innovating the future. Imagine a world where breaking a hand means nothing, a world where you can still have movement even after losing a limb. Well, this has been the aim of prosthetics for since its start. Over the past few centuries, prosthetics have had a significant impact on the lives of many individuals. It enables people with physical disabilities to have a greater quality of life. And with the introduction of neuroprosthesis, like neuromodulation, the possibility of recreating hand movements, even in a paralyzed person, is getting closer to reality. But there are still some glaring problems, and today I'm going to go through the different types of neuromodulation and analyze their failures. Hi, my name is Anirudh, and I live in Folsom, California. I'm a junior in Folsom High, and I love to learn about neurology and do research about it. Some of my hobbies include robotics and also playing tennis and basketball. I'd also like to thank my mentor, Dr. Riggins, for guiding me through this process. So, why did I choose neuromodulation? Well, neuromodulation is a culmination or mixture of both robotics and neurology, two of my favorite subjects. It also provides a solution to many problems in the medical field, like Parkinson's disease, Alzheimer's, and SEI. So, what is SEI? SEI stands for spinal cord injury, and it's caused by a variety of reasons, including vitamin deficiencies, accidents, falls, and many more. And so there are two types of SEI, complete SEI and incomplete SEI. Complete SEI refers to the permanent damage that occurs to the spinal cord, and this can affect the bones, the nervous system, the blood vessels, and many more. Whereas incomplete SEI is temporary damage, as it preserves motor and sensory functions, and they can be healed over time. And there are two phases to the SCI, primary phase and secondary phase. The primary phase refers to the physical injury that happens to the spinal cord itself, and if it affects the surrounding area, including the bones, the blood vessels, and the nervous system. Whereas the secondary phase refers to the biological and chemical processes that spread and worsen the injury. And SEI is prevalent and is a big problem in the medical field. And how do we solve this? Well, this is where neuromodulation comes in. And the first type of neuromodulation device are transducers. Transducers work on the concept of stimulating the part of the nervous system that needs to be that has been disabled. And this can be done through a variety of mediums. And the first one is electrical transducers. Electrical transducers use electrodes and deliver an electric shock to the targeted part of the nervous system. Whereas mechanical transducers, they alter the structure of the cell membrane, in turn activating the cell. Chemical transducers, on the other hand, are completely different, as they release chemical molecules into the brain, which activates the peptide receptors instead of the neurons. Transducers alone can be used directly into the spinal cord to work, and they need to be implemented into another device. And epidural electric stimulation is such a device that uses transducers. It also can be said EES for short. EES is a form of therapy that brings back volitional or voluntary control back to the spinal cord. It works through activating specific parts of the spinal cord that has been disabled. And now is the perfect time to say that there are four sectors of the spinal cord: cervical, thoracic, lumbar, and sacrum. And activating or stimulating different parts of these sectors leads to different results. For example, there was an experiment done on cats where activating the regions of lumbar 4 and lumbar 5 region of the spinal cord led to back limb control, but the exact same results can be replicated in humans, and hence the same thing had to be done again. And the following table shows some of the results. By using the frequency range of 25 to 60 Hz in the spinal cord sector cervical 7 or C7, the effect was that it enabled standing, stepping, and voluntary leg controls. Whereas using the range of 30 to 60 Hz in the spinal cord sector T11 to L1 led to enabled bearing of complete body weight, which also means that the patient was able to stand properly without support and without damaging his or her spinal cord. EES also enables verbal, auditory, and visual function. And keep in mind that in the table, the frequency is a range instead of like a single number. And this is because the anatomy of different humans varies slightly, and this causes a major difference in the frequency. We'll cover more about this in the further slides. Although EES is pretty effective, there are still some glaring problems. For example, EES can be used alongside MRI, as the electrode tip gets heated when the radiofrequency waves of the MRI interfere with the EES device. And hence, an alternative to this is magnetic stimulation. Magnetic stimulation uses microcoils and magnets instead of electrodes in the case of EES. They are also much smaller than the EES devices, as they're only 3 millimeters in radius and 3 millimeters in height, when compared to the other device that's about 2 to 3 inches in all directions. These can also be turned off to prevent interference with other devices like MRI, ensuring more longevity of the device. In order to prove that micromagnetic stimulation, or just magnetic stimulation, is effective, they were placed near retinal cells. And this is because retinal cells show stimulation or spikes to both light and electricity. And so when similar stimulation was shown to light and this device, it was shown that micromagnetic stimulation provides a near natural response by the body. Although neuromodulation devices have been improving over the past few years, and implementation of neuromodulation is not an easy feat. An issue comes at the very first step, surgical implantation. So one method through which electrodes are implanted into the body is laminectomy, which is a biomechanical process that poses many risks, such as spinal cord instability and also spinal cord deformation. Implantation surgeries are also a lengthy process because the laminectomy damages the tissue surrounding it, and which leads to a severe damage to the tissue, which requires 36 months to heal. Another problem with neuromodulation is the size and comfort factor. Although they have been getting smaller over the past decade, they still are pretty large and cause damage to the blood vessels and the neural system surrounding it. These devices struggle to reach their target because of tissues too, and that's the third problem. This is also known as tissue heterogeneity or tissue impedance, and this was the reason why the frequency was arranged in the table we looked at before. So there are many properties of tissues that provide different resistances to the electrode, and it makes it extremely difficult for the device to reach the intended target with the desired frequency. This process is further worsened by neuronal activity, as the stress causes the resistance in the tissues to be worse and even varying. And all these problems are caused during surgical implantation, as when the device is implanted, the immune system of the body recognizes the electrode as a foreign body and leads to a foreign body reaction where microglial cells are released and they form a thin and cellular sheath which covers the electrode. And this leads to many problems because the electrodes now can't record data or provide stimulation. In order to prove that the electrodes really affect the brain in an invasive way, nanofluorescent beads were placed in the blood vessels, and they were observed to be leaking near the blood-brain barrier in the exact place the electrode was implemented. And keep in mind that no damage was observed prior to the implantation, further proving that neuromodulation is a really invasive process. We've seen so many problems. So how do researchers categorize them? So how do they make it easy to recognize what type of failure it is? Well, some professors at the New Jersey Medical School have performed a retrospective study, which means that they were analyzing an experiment that occurred in the past. And they've made two categories. The first is by type. There are four groups in the categorization of type, which is biological, material, mechanical, and unknown. And each of these have different problems, as biological refers to the foreign body response, material and mechanical refer to the circuitry problems, and unknown refers to the loss of recording data. And the second type of categorization is by time. And the first type is by acute, which refers to the rapid effect loss of effectiveness and signals in the period of 7 to 8 days, whereas chronic failures refer to the loss of signal over a longer period of time, ranging from a singular month to many years. The following chart shows the different failures in neuromodulation when related to the acute failures. So the most amount of failures are found in mechanical, suggesting that there needs to be further development in the understanding of the circuitry of neuromodulation. And while looking at the chronic failures, there were only 9 biological, 2 material, and 6 unknown failures. There were no mechanical failures at all. This seems to suggest that the mechanical failures show themselves in the first 7 to 8 days, and if you don't observe any failures during that period of time, that means that the circuitry of the devices are great and they don't need to be tampered with. So what's next? What do we need to see developing in the field of neuromodulation that would take it to the next level? But the first and the most crucial aspect is having a deeper understanding of the neural anatomy. And we've seen many problems that interfere with the device, like tissue impedance, microglial cells activation, and many more. So having a deeper understanding of the human anatomy would help realize scientists how to counter and work around these problems. A second solution that needs to be worked on is the introduction of closed-loop neuromodulation. As you can see in the picture below, there are two types of neuromodulation, open loop and closed loop. Open loop refers to the device that only stimulates the brain, whereas closed loop refers to the device that stimulates the brain and records and decodes data too. It's similar to AI, which records its own stimulation and tries to improve in its next cycle. So what are scientists working on currently, and what are the future efforts? Well, the first is scientists are working on altering the shape and size of the electrode. They're coming up with a thin and sharp lattice-style probe that has a low surface area, so it can uh, be implemented into the brain without much damage. And this leads to lesser probability of the microglial cells activating. The second solution is stem cell transplantation. As you've seen throughout the presentation, we've seen that the glial cells provide a great problem to neuromodulation as they cover and prevent the electrode from working. So how do we ensure that these cells are disabled for a short period of time? And that's where stem cell transplantation work comes in. This process resets the immune system and disables them for a short period of time, the period of time it would take for the surgeons to implement the electrode. And this is already prevalent in treating other problems like sclerosis, so it doesn't pose much of a risk and would be a great solution. Conclusion. So I believe that neuromodulation is a growing field of medical treatment and provides a solution to many neurological diseases. It provides aid to some of the most quintessential components of the body, like the spinal cord and the brain, either through micromagnetic stimulation, epidural electric stimulation, or any other form of neural transducers. However, more work needs to be done to solve additional issues, as this can only be achieved through expanding on the knowledge of the interaction between cells and the implant. And this not only requires the field of biology, but physics, biochemistry, computer science, and many more subjects to be implemented into the field of neuromodulation. That concludes my presentation. Thank you. Any questions?
Thank you so much, Anirudh. Let's see here. So there is a question about, you had mentioned AI, and I'm curious to find out, in your research, what did you hear about the possibilities or read about the possibilities of using AI in these applications? And do you have any concerns about that?
Yeah, so I've seen many articles referring to AI and neuromodulation. So what basically happens is that once the device activates the brain, it records data. So it sees how the brain's working and how efficiently it's delivering the electricity. And after decoding this data, it realizes through what means should it deliver the electricity and kind of improves its cycle and does better next time.